Good afternoon, everyone, and welcome, Ms. Alena Semkina. I am Dr. Shyam Prasad Sajankila, Associate Professor in the Department of Biotechnology. And since Dr. Ujjwal is held up in a plenary session, she may be joining us a bit later, and I'll be with you during the proceedings. We are in for the third lecture, keynote lecture for the series, which is presented by Ms. Alena Semkina. She is a Deputy Director, Chief Commercial Officer at VG Khlopin Radium Institute of Rosatom Corporation, St. Petersburg, Russia. And she will be talking on the biotechnologies in radiopharmaceuticals, development by example of recombinant alpha fetoprotein as a vector in targeted radionuclide therapy and diagnostics. She has immense amount of experience in the field of radiopharmaceuticals and targeted therapy as well as diagnostics. With this brief introduction, I request Ms. Alena Semkina to take over the stage. Over to you, ma'am.
Hello, can you hear me
Hello.
well?
Yes, we are able to hear you.
Okay, good. I'm sorry I was a little bit late. Can you share my presentation which I just sent to the email which I received from Dr. Ujwal? Sorry if I pronounce his name in incorrect
No,
form.
yeah, it's right.
Or shall
We
I
will
do it
sure
myself?
we'll do that. Just give us a couple of minutes, ma'am. ಉಜ್ವಲಿಗೆ ಕಳಿಸಿದಾರಂತೆ PPT.
Also, maybe I can try to do it myself if
I think that
that
would be
is.
better. While we search, I think that would be a better option also.
Could you please guide me? Shall I just put the bottom to start
There
showing,
is a,
right?
there is an up arrow button in the. I mean, once you open the PowerPoint, there is a. Okay, we are accessing the presentation, ma'am. Just a minute. We will present it for you. We are in the process of downloading and presenting it. If you can give us a minute.
Maybe I can start some introduction information.
Yes, I think that would be better. We are presenting it in just a minute.
Yes.
Yeah.
The first slides of my presentation will be anyway connected to some general information, so I can start, I think.
Okay, ma'am. Just go on with that. Thank you.
Thank you, and thank you for this opportunity to make this report. Actually, I'm not a specialist in biotechnology or biochemistry, so please accept my apologies if I make any mistakes. Please feel free to correct me. I would be just very glad. Our institute is specialized in different spheres connected to radioactivity and its application for different purposes. This is Radium Institute
I
named.
think the presentation is now being shared, ma'am.
Yes, yes,
Are
yes,
you
I
able to
can
see
see
that?
it.
Yeah. Yes, yes. Thank you, ma'am.
Thank you very much.
Yeah.
So our institute, Radium Institute, named after a great scientist, Mr. Khlopin, was founded by academician Vladimir Vernadsky. He is a very famous scientist, well known all over the world, and he expected radioactivity to have a lot of different application and to serve for common good in future in absolutely different technologies, and especially with the power that is hidden inside of the nuclear nuclear of the atom. So Vladimir Vernadsky uh, founded our institute 99 years ago. So next year, that will be 100 years of our institute. And in our institute, there are several departments or divisions which are dealing with absolutely different applications of radioactivity. So in our institute, we are making research of fuel cycle and fission technologies. We are making research in isotopes enrichment, including radioisotopes enrichment. We are running different R&D and investigational work in radiochemistry, geochemistry, radioecology, and also there are several production departments and department of radiopharmaceuticals development and production. And recently we started absolutely new work for us, which is connected to the sphere of biotechnologies. So I will make a short description of the approach. I'm not sure if all of you work with radiopharmaceuticals, and if all of you know which tool can be founded for biotechnology using different radioisotopes. And the first slide, how can I go to the second slide, or can you help me with this? Yeah, thank you. So I will make a general description of this tool, which I spoke about. This tool that help physicians and scientists to understand human body much better, and to see how our tissues, how our organs functioning. What I mean, there are two main applications of radioactivity for medicine. Of course, there are a lot of other applications. I will not talk about them today. I will speak only about one particular question. So, two main application which I'm going to discuss today. There are diagnostics and therapy. And when we speak about diagnostic, radioactivity provide us unique solution. What means unique? It means that we do not see a morphological picture of our tissues. We can see how it work, if it is alive or not. What is the metabolism? Does it have any problems, any disorders, or maybe specific behavior? This can be shown only with help of radiodiagnostics. Another application is radiotherapy. That is actually not that radiotherapy, external radiotherapy, which we can find in many hospitals. When we speak about external source of radioactivity, it can be gamma irradiation, it can be proton irradiation, and which is used for cancer treatment, for example. We speak about radiotherapy, which is target radiotherapy, or sometimes it calls nanochirurgy. So we speak about molecular therapy, molecular diagnostics, and molecular therapy. So how can we construct this tool to see on the molecular level the function of our organism and our tissues? How can we treat it on this level? We should construct a molecule which can specifically come to particular tissue, to particular cells, and radioisotope which is connected to this molecule can show us, if this is diagnostic, it can show us the localization of this tissue, or even a very small amount of cells which cannot be located with other methods. So, for example, very small metastases can be detected only with radiopharmaceuticals, only with this precise method of molecular imaging. So when we construct this molecule, which can specifically target the cell, or when we try to destroy the cell, this particular cell, we should take into account 2 points. We should choose the best radionuclide, and we should choose the best vector for these purposes. I'm not going to describe all this range of radionuclides which are used and all their characteristics which help us to understand which radionuclide can be used for which purposes. I just tell in general that there are a wide range of radionuclides, and all these years, maybe roughly 80 years of application radioactivity for medicine, there was type of change from one radionuclides to other radionuclides, and it was type of natural selection of radionuclides which are best for nuclear medicine. And if any of you are interested in this information, I can provide you more details. Please feel free to write me, and that's not a problem. Because I personally think that this is perfect tool, and this is perfect tool for biotechnology, definitely. So if we speak about radiotherapy, this is vectorized radiotherapy. It means that our molecule should specifically target that cells which we are interested in, should specifically locate there, and should be accumulates there. Also, it should be stable, and we should be sure that our radioisotope will be delivered to the cell, for example, cancer cell. We should be sure that this molecule doesn't destroy our organism as well. So there are several approaches how we choose these molecules. There are metabolic radiotherapy. For example, we can use iodine, and this is one of the most famous radioisotope which is used for radiotherapy. Of course, this is because of its metabolic characteristics and accumulation in thyroid tissues. So also there are other methods like radioimmunotherapy, when, for example, antibody or maybe fragment of antibody is used as a vector, and this vector delivers our radioisotope to the specific cancer cells with specific receptors. And this is very popular mechanism, which is now under investigation in many countries, and clinical trials are running in many countries right now. Also, there is peptide receptor therapy, which is also very popular, and I will speak today a little bit about it. So, mostly as vectors, there are used antibodies and fragments, peptides, aptamers, scaffolds proteins, and small molecules. Different molecules. Actually, there are a wide range of molecules which people try to use as vector to deliver radioisotope. There are a lot of complications of these technologies. I noticed in this slide this complications, but that benefits for patients, of course, are the most important. So, what is per my understanding, per my view, what is the most important benefit for patients? The first, that is, of course, efficiency. This is extremely efficient compared to chemotherapy or even other methods. Extremely efficient method. Very few side effects, and not strong. There is other very important benefit. This radioisotope, which is used for radiotherapy, it's type of toxic agent for the cells to destroy them, and this type of toxicity is invisible for cancer. What does it mean? If we speak about drug resistance, if we speak about genetic reprogramming of cancer cells, when we use conventional or even target chemotherapy, anyway, our cancer cells definitely would need more and more strong and more toxic drug, with each next line of therapy. That is not necessarily for radiotherapy. There are a lot of questions which do not still have answers, but probably that is because of microenvironmental changing, maybe because of some other effects. But cancer cells sometimes disappeared completely after several treatments, and there is no problem like more and more aggressive cancer after each treatment. Okay, I will show you the pictures about this situation, which show these results. Radionuclides, which are used and shown on this slide, can be absolutely different, and there are a lot of methods to produce them. And for example, in our institute, we use all of these methods, and we have a wide range of different radioisotopes for different applications, not only for radiopharmaceuticals or for biochemistry. Mostly, we use them for other industrial applications. There are approaches when you can use same chemical element for diagnostics and for therapy. For example, like I told you about iodine, or there are other elements which can be used in the same way. I mean that if we can use iodine 131 for thyroid treatment, we can use iodine 123 for diagnostics. And we can see the result of our treatment, or we can see it and watch it with very good understanding of what is going on inside of the organism. If we label our molecule, biological molecule, with other radioisotope, for example, scandium or terbium or other radioisotopes, we can also construct two molecules for diagnostics and for therapy, which can work in the same way and target our tissue or some cells which we are investigating, and then treat these cancer cells if we speak about cancer. Can you please move to the next slide? So that is the result. One of the methods, peptide receptor radionuclide therapy. Now this drug is that was acquired. This company was acquired by Novartis Corporation, and now this drug received marketing authorization, as you see, in Europe 4 years ago, in the USA 3 years ago. You can see the result. If we speak about some conventional standard approach, that is absolutely completely different result, completely different efficiency, completely different side effects and everything. I didn't include here a photo of patients, but these photos which shown by the doctor who was a pioneer of this peptide receptor radionuclide therapy impress a lot. So very complicated patients. You should understand that this is usually that is metastatic cancers and very bad patients, and this complete response, which happens rather often, impress a lot. So there is nothing which can be compared right now. So that is the first drug, the first radiopharmaceutical of this type, which has already received approval in many countries. Can you please show the next slide? That is another example. This is PSMA targeted radioligand therapy. So it's used for advanced metastatic prostate cancer. And you can see these photos of the patient. That is photos made with help of nuclear medicine technologies. So you can see the first photo, and you can see metastasis on these photos. On the first photo, there are PSA on the level 4.6. The second photo, you can see same patient 1 year later, and this patient received standard treatment, conventional treatment, and you can see how many metastases he had in his body. So after this situation, when PSA became 45, he was recommended and he was treated with 2 cycles of lutetium-177 PSMA targeted radioligand therapy. And you can see the result on the third picture. So this is complete response, and this result is rather often for this application. Next slide shows another picture. Could you please? Yes, thank you. On the previous picture, that was lutetium therapy, and lutetium is a beta emitter, so it's emitting electron during during treatment. And here you can see targeted alpha therapy, where used actinium-225 instead of lutetium. And on the first line, you can see 3 pictures, and you can see metastatic prostate cancer, which was treated with actinium PSMA, and you can see the result. So this is complete response. And the situation with lutetium PSMA, when lutetium PSMA therapy did not work, and after this complications with lutetium therapy, there was used another radionuclide with the same vector, and you can see the result. So complete response as well. There are a lot of such examples, and this therapy is used all over the world. Probably all of you know about this. So I'm sorry if that is something to repeat for you. But I can say that in many countries, many doctors, many biotechnologists do not know about these methods at all. Could you please show the next slide? The most problem is that for the examples which I noticed before, there were actually 2 applications: neuroendocrine tumor and prostate cancer. But there are a lot of different type of cancers, of course, and there is a dream to find some universal targets, and of course universal vectors, which could help to target it. So on this slide, you can see a photo which was called the image of the year during SNMMI conference. It's international conference in the USA. And you can see this result when was used the fibroblast activation protein inhibitor, FAPI. And this radio tracer, single radio tracer, can be used to diagnostics a lot of types of cancer. In their publication, they say it's nearly 30 types of cancer. Of course, there is a very big interest if this vector can be used for treatment, for therapy, or not. This question means that there should be done a big research of stability, of biodistribution in vivo, and of course, when we speak about therapy application, it's more complicated and it's more expensive compared to diagnostics applications. And finally, what we are doing now in our institute. Can you please show the next slide? This is the work which is done together with several biotechnological companies. This is a investigation of alpha fetoprotein, a well-known protein. Investigation of this protein as a vector to target different types of cancer. So what is promising? First of all, there are a wide range of cancers with overexpression of receptors for alpha fetoprotein. This information is well known, and this protein is well known for many years. So this is something understandable, and the only one issue which have been a problem, a headache for many years, is that there was no one high-scale production of this protein in the world. So there are several recombinant alpha fetoprotein manufacturing sites, but the self cost and the price of this protein is extremely high. Of course, there is a source which is natural, not recombinant, but it's also limited, expensive, and not pure. Also, there is a other aspects which I mean ethical aspects as well. So what changed in the recent time, in the past years? Right now, there are two independent methods of high-scale production of alpha fetoprotein in Russia. And as we know, as we read in publications, there are another method which is used in China. And these people also receive very promising results investigating this protein as a vector. What is going on in Russia right now with this protein? There has been two independent, not connected researches. One is about application of alpha fetoprotein for wide range of diseases not connected to oncology. But if we speak about our application, since we plan to use alpha fetoprotein for cancer treatment, we concentrate on its properties as a vector for cancer. And we received, not our institute, but the group which we are working with, received very promising results in preclinical trials, and clinical trials have also started. So the proof of concept when alpha fetoprotein can be used as a vector for toxic agent. In that preclinical trials and clinical trials, as a toxic agent, there is the use of actinomycin. Sorry if I pronounce it not in correct way in English. Actinomycin, cytostatic composition, and absolutely in the same way, with of course several differences connected to radioactivity. We are now running trials to synthesize a molecule where alpha fetoprotein is used as a vector and radionuclide is used as a toxic agent. So all the results which are received right now in experiments in vitro, which we do not consider as a very interesting results, but also in vivo, which we consider as interesting results, shows, I may say, promising applications of this vector. And right now there are several non-solved questions, non-solved problems, like complicated labeling, connected also to complicated quality control of the final molecule, stability studies. I mean stability studies when you should be sure that your vector is able to deliver a toxic agent as a radionuclide to the particular cell. And of course, there is another question which is also complicated. That is the question of other tissues, not cancer cells, but other cells, which also have receptors for alpha fetoprotein. So that is the problems that we are investigating and solving in our research. And we would be glad if any of your specialists is interested to participate in this research. So I tried to make it as short as possible. I'm very sorry if that was not clear for maybe some reasons. And if you have any questions, or maybe if you are interested to receive any additional information, please feel free to contact me. And there is my friend, Dr. Afsal, from SMT Healthcare. So he is very good radiopharmacist who, of course, can answer some other questions, of course, connected to this problem.
Thank you, Ms. Alena, for that wonderful talk. We have a couple of questions for you. If I could put it up. First one is, could the radioisotope itself have an effect on the molecule after being labeled?
Yes, of
Suppose the
course.
yeah.
Yes. That is one of the main problem. Um, if you use such radioisotope like iodine, you can easily connect this radioisotope to mostly maybe to all biomolecules. And if we speak about other radioisotopes like metal radioisotopes or other with another chemistry, you would need to use probably chelating agent. You would need to use linker, and of course, this construction makes influence on the biomolecule, and this is one of the most important and most complicated question. So you should be sure that your diagnostic molecule modified with radioisotope have has same biodistribution as therapeutic molecule.
Okay. Then we have the second question. Uh, what about the nephrotoxicity of radioisotopes which are being used in the diagnostics or therapy?
Both these questions were notified in my presentation. I didn't stop on this question. If you go to the probably that is the first slide.
Okay.
Um, yes, that is the first slide. So you can see here um, there is a problem which should be taken in account concerning renal and liver uptake. So if we speak about nephrotoxicity, it means that this radioisotope, sorry, not only radioisotope, all the molecule has out sorry, I don't know how to say it in English. So it has renal uptake, and this radiopharmaceutical can damage and can have nephrotoxicity. So one of the important question is to use molecules which would have not renal but liver uptake. And when we speak about liver uptake, in this case, um, that would be the better way, and there is no nephrotoxicity in this case. And this, of course, is taken into account for many people who are developing radiopharmaceuticals. So you are correct. This is important question, and it is always under consideration when you develop new radiopharmaceutical.
Okay. And the last question is, what would be the criteria for choosing the best radioisotopes for a particular approach, diagnostics or therapy?
I'm sorry, I didn't understand the beginning of your question. What is
What would be the criteria for choosing the best radioisotopes for the therapy or
criteria?
diagnostics? Yeah.
Yes, I understood. Criteria. You can see on the first slide as well. There is written type of radiation, energy, and lifetime. That is all these are criteria for radioisotopes and chemistry. So if we speak about chemistry, that's your first question. If radioisotope modify or non-modify the molecule. Um, when you use iodine, this is um, the way how to reduce modification, how to minimize this influence. But if we speak about other radionuclides, that is another chemistry. So that is the first criteria. If you can use, if this chemistry change your biodistribution, your specificity, or do not or does not change. Another criteria is type of radiation. If you want to make diagnostic molecule, you should use gamma emitters, or you should use positron emitters. If you use gamma emitters, that is SPECT technology. If you use positron emitters, that is PET technology. Of course, very important is lifetime. It should be the best for your case. It should be adapted for biodistribution of the molecule. For example, if you have big molecule like antibody, you should take radionuclide, which half-life roughly 2 days or 3 days or even more. If you take small molecule, you should take radionuclide which live much shortly. Yes, so it has a shorter lifetime, but also very soft soft radiation because of this renal uptake and nephrotoxicity. So if this is very soft, there is no problem for renal at all. And if you need a radioisotope for treatment application, of course, you should use electron emitters, beta emitters, or alpha emitters, which are even better. And for therapeutic application, you should also understand all these complicated things like lifetime, the energy, and possible renal uptake. So that is all these criteria are taken in account to understand what is the best way.
Okay. Thank you so much, Ms. Alena. I request Dr. Ujjwal to take over the session now. Over to you, Dr. Ujjwal.
Yeah, very good evening, Miss Alina.
Good
I'm
evening.
I'm delighted to have you, and we had a good interaction. Your talk was really enriching. Dr. Afzal, would you like to add something? We have industrial experts.
Thanks, Ujjwal. We don't have it because this is a very good presentation. Thanks, Alina, for sharing your inputs to Nitte-Bio group. Definitely, nuclear medicine connected to biotechnology is a growing field, and it is something like we are expecting the researchers to investigate more in this aspect. There are many questions. I heard all the questions and Alina answered the question. So we would like to share more knowledge regarding nuclear medicine with the Nitte students and Nitte team anytime. You all can approach us. Our contact details are there with Dr. Ujjwal. So we are happy to help you all. Thank you.
Thank you, Alina, for all your support and your enriched guidelines and your talk. I am very delighted to have you for this conference. Thank you once again from the team of Biotechnology Engineering NMIT Nitte.
Thank you very much. Thank you.
Thank you. Thank you. Have a good day.
Thank you
Have a
all.
good day.